This is a bacterial cell, E. coli to be specific. We are going to look at it as an example for the central dogma. First, let's slice it open to see inside. Bacteria are fairly simple as organisms go. They are prokaryotes, meaning they have no membrane-bound organelles. They possess a cell wall to give them shape and structure, a plasma membrane, like all living cells, to separate their internal workings from their external environments, ribosomes for assembling proteins, plasmids for storing and exchanging genetic information, and a circular chromosome, which stores the bulk of the genetic information for the cell. This cell also possesses a variety of other proteins, molecules, and biochemicals, but they're unimportant at this time. Before we can question the central dogma, we need to understand what it is. We need to understand the steps involved. In general, the central dogma is the flow of information from the order of nucleotides in DNA to the order of nucleotides in RNA, and finally to the order of amino acids in a protein. This is a complicated process involving many steps to get from DNA to protein. Now that we have the basic idea of the flow of information, we can look at the central dogma in greater depth. Here we can see the bacterial chromosome. It is circular, unlike our own, which is linear. Here we can also see some accessory proteins, the clusters of 4 dark green spheres. These help to pack the DNA into a smaller space and keep it organized. The chromosome in bacterial cells is free in the cytoplasm, unlike us, where ours is contained in a membrane to form a nucleus. Let's make this a little less cluttered before we go on. Here we can see the characteristic double helix of DNA. It consists of a phosphate and sugar backbone. Between these two ribbons are the nitrogenous bases. Although this is just a cartoon model, it will suffice to illustrate the point. There are 4 different bases in DNA: adenine, guanine, thymine, and cytosine. Adenine and thymine always pair together, while cytosine and guanine always pair together. Adenine and guanine are purines, while cytosine and thymine are pyrimidines. Both strands are held together by hydrogen bonds between the bases. Adenine and thymine share 2 bonds, while guanine and cytosine share 3. These bonds hold the strands together, but not so tightly that the strands can't be separated to read the bases. Let's zoom out a bit to see the parts of a gene for a protein. Here is a gene in cartoon form so we can better outline the important parts. First we see the promoter. This is recognized by a group of proteins that are used to copy the DNA into RNA. We'll discuss this more in a bit. Next we see the ribosome binding site. This component is translated and allows the ribosome to bind to the mRNA and read it. The coding sequence does as its name suggests. It possesses all of the information about the sequence of the amino acids in the protein. Finally, there is a terminator sequence that stops the RNA transcription process and essentially ends the mRNA. All of this is needed to get information from DNA to mRNA and have the mRNA formatted for being translated into a protein. To start transcription from DNA to RNA, we start at the promoter. A number of factors recognize the sequence as the beginning of a protein. These factors help RNA polymerase become associated with the DNA. They can also regulate, along with the promoter sequence, the expression rate of the protein. Here we can see how RNA polymerase begins transcription of the DNA. The polymerase continues transcribing DNA to RNA until it hits the terminator region. When it does, the mRNA folds quickly and pulls the mRNA out of the RNA polymerase, terminating transcription. This causes RNA polymerase to stop and fall off the DNA, to be reused by the cell. Now, the RNA is free and contains all of the protein coding information from the DNA. We can see that DNA and RNA are very similar, and indeed they are. The only major structural difference is that RNA is single-stranded, while DNA is double-stranded. The sugars that make up the backbone are also different. One of the defining features of RNA is that it contains uracil as a base instead of thymine. Uracil may be slightly different, but it still possesses the preference to bind to adenine. Here we can see the details of the mRNA. We can see that it has a ribosome binding site, RBS, and a coding sequence. The RBS allows and promotes a ribosome to interact with the RNA to produce a protein. The coding sequence possesses the information about the sequence of amino acids in the protein. There are 2 other important features to point out: the start codon and the stop codon. The start codon assists in binding the ribosome and marks the beginning of the protein. is always the amino acid methionine and represented by the AUG codon, at least as far as we know. The stop codon is what ends the protein. This is one of the three codons UGA, UAG, or UAA. These cause the protein to release from the ribosome and for the ribosome to dissociate from the mRNA strand. Here we can see the start codon in better detail. For the mRNA to be translated into a protein, we need to have a place to start and a way of assembling it. To do this, a tRNA carrying a methionine amino acid must interact with the mRNA. This interaction is brought about by the complementary base pairing between the codon, or group of three nucleotides on the mRNA, and the anticodon complement of the tRNA. When the methionine tRNA binds, it also aids in the binding of the 30S ribosome subunit. This is only part of the ribosome, but it will help the 50S subunit to bind and form a complete ribosome. The 50S subunit attaches to the mRNA complex, completing the ribosome. Now the production of the protein can begin, one codon at a time. The methionine tRNA currently sits at the center of the ribosome, the P slot. The next tRNA carrying the next amino acid enters at the A slot. This tRNA is carrying the anticodon for the next amino acid. Once this tRNA has entered the A slot, its amino acid is bonded to the methionine with a peptide bond. This frees the methionine from its tRNA. Now that the growing protein, or chain of amino acids, has been transferred to the new tRNA, the ribosome transits and shifts downstream by one codon. This moves the methionine tRNA to the E slot, where it is ejected from the ribosome. This tRNA will be recharged with another amino acid and reused by the cell. The tRNA formerly in the A slot is now in the P slot. The ribosome is now ready to receive another amino acid. The cycle of transferring the chain from one tRNA to another will continue until the ribosome reaches a stop codon. Ready, set, go. Here we can see the ribosome working a little slower than actual speed. When the stop codon is reached, the growing polypeptide, aka the protein, is released from the ribosome. At this time, the components are also separate from the mRNA. Now the ribosome components can be reused to make another protein. The mRNA can also be reread to create more protein. We now have our polypeptide chain, or chain of amino acids. Each amino acid represents a codon that was read. But what does it all mean? Why did the codons need to be 3 bases long? Let's answer these questions. For starters, we need to do some math. There are 20 amino acids used by organisms. Each amino acid has the same basic backbone, which is why they can be bound to form a protein. Each of the 20 amino acids has a different functional side group. These are what cause the protein to fold and do what it does. Here's where the math starts. We have 20 amino acids, but only 4 nucleotide bases. If the bases A, T, C, or G were to represent an amino acid, then we would only have 4, far too few combinations. If each amino acid were represented by 2 bases, that would mean that there would be 16 possible combinations. We would not be able to represent all 20 amino acids. What about a combination of 3 bases? This would give us 64 possible combinations, more than enough to represent the 20 amino acids and stop codons. But is it too much? No. Many of the amino acids are represented by multiple codons, often very similar codons. One benefit of this is protection against errors in translation. Now back to protein folding. Once we have the primary conformation, i.e. the linear chain of amino acids, it can fold into its secondary conformation. This step is dependent on the side chains interacting. The alpha helix is a coil formed by hydrogen bonds between parts of the backbone of the polypeptide. This is one of the basic structures of a protein. The beta-pleated sheet is formed again by hydrogen bond interactions between parts of the backbone and the polypeptide. It is a sheet in the sense that you have segments running anti-parallel or parallel to one another. Depicted is a cartoon model representation of these structures. The protein can then fold into its tertiary structure. This is primarily driven by interactions between side chains on amino acids within the protein and between the side chains and the environment. These interactions cause the protein to achieve its final shape. Now that the protein is folded, it may be modified. This could mean having segments cut out, side chains modified, or disulfide stabilizing bridges formed between cysteine side chains. Once the protein is ready, it can do its intended job. Let's recap. The central dogma is about the flow of information from DNA to RNA via RNA polymerase, and then from mRNA to a protein via a ribosome. This process is one of the most important to life on Earth, and is complex, and hopefully it now seems a little more manageable.